and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing West Bengal set exam question paper for the subject life science. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of WB set exam being discussed, you can go to my channel Omni Bio Entrance. In the channel playlist, select for WB set and you will find them there. So coming to the next question. Light inducible seed germination termed photoblastism occurs in some higher plants. Which one of the following is an example of positive photoblastic seed that does not germinate in darkness but requires exposure to sunlight? So photoblastism, it is a condition where seeds require light to germinate and they will not germinate in darkness. That is, they require light for initiating germination. So they have asked positive photoblastic seed that does not germinate in darkness but require exposure to sunlight. So out of these, the answer is lettuce. Answer is option A. Then next one. Density gradient centrifugation of genomic DNA of a specific organism revealed a lower density peak in addition to the main peak. So this is due to, so density gradient technique, it is used to separate DNA molecule based on their density. So AT rich regions are less dense than the GC rich regions in the DNA. So the presence of lower density peak in addition to the main peak in a DNA profile of an organism, it suggests that the molecules are AT rich. Okay. This lower density peak in addition to the main peak. So lower density peak, it suggests the organism as AT rich. So the answer here will be AT rich satellite DNA. And this AT rich DNA sequence, it is found usually in the centromeric and telomeric regions of the chromosome. And satellite DNA, it is a type of DNA sequence that is repeated many times in the genome. GC rich satellite DNA, then it would have resulted in a higher density peak, not a lower density peak. Then median of first 100 natural numbers. So the first 100 natural numbers are your 1, 2, 3, 4, 5, 6, 7 up to 100. The 50th number is 50 and the 51st number is 51. So you have to take the median as the average of these two numbers. So 50 plus 51 divided by 2 will give you 50.5 as the median. Then if the slope of the regression line is 1.75 and the intercept is 12, what is the value of y when x is 3? So y is equal to mx plus b, that is the equation. So you have to, y is the value of y, m is the slope, x value of x, b is the intercept. So you have to find the value of y when x is 3. So substitute the values, you will get it as 17.25. Answer is option C. So coming to the next question. In In C4 plants, refixation of carbon dioxide occurs in chloroplast of. So in C4 plants, carbon dioxide fixation, it occurs in two stages. So in the mesophyll cells, carbon dioxide is fixed into 4-carbon compound by the enzyme phosphoenol pyruvate carboxylase. Then this compound, that is the 4-carbon compound, it is transported to the bundle sheath cells and it will release carbon dioxide, and this carbon dioxide is fixed via the enzyme Rubisco. So here refixation of carbon dioxide occurs in the chloroplast of bundle sheath cells. Quiescent centers of root epiphysis serve as. Their answer is option C, that is reserve block of diploid cells when the root tip is damaged while pushing through the soil. That is the answer. Then next one, which of the following statement is incorrect? Root apical meristem is the major site of the synthesis of free cytokinin. Yes, that is correct. Amino acid methionine is the precursor of ethylene. That is also correct. Benzyl adenine is the most abundant naturally occurring. It is not naturally occurring. It is a synthetic cytokinin. Okay, so that is the incorrect statement. Tryptophan is the precursor of IAA. That is also correct. Then next one. During secondary wood formation in plants, balloon-like protrusion called tylosis are generated from. So tylosis are balloon-like protrusions that form from the xylem vessels of woody plants during secondary wood formation, and they are generated from the axial and ray parenchyma cells. So answer is option A. Then in which of the following chromosomes lateral loops of DNA are present? So it is the characteristic of lamp brush chromosomes. So these loops are formed when the chromosome is undergoing transcription. And these lamp brush chromosomes, they are found in the oocytes of certain amphibians and birds. Then B chromosomes, they are found in some organism and they are small extra chromosome and they do not have lateral loops. Then sex chromosomes, you know, they are used to determine the sex of an organism and is not involved, does not have lateral loops of DNA. Lagging chromosomes that are delayed in their movement to the poles during cell division. Incertae sedis is the term used for a taxonomic group where its broader relationships are unknown or undefined. Answer is option C. Its phytogeography is not known. Phytogeography, it is the geographic distribution of plants. That is, you study the geographic distribution of plants. So Incertae sedis is not involved there. Its characters match with a particular endemic species, and endemic species, they are found in a particular geographic region. So it is not related there also. Its distribution is found in the Galapagos Island. That is also not correct. So answer is option C. Identify the wrong statement about bulliform cells or motor cells. So they have asked for the wrong statement. So these are subepidermal cells that occur on the they are the found in the epidermis of leaves and found typically on the adaxial, that is upper side of the leaves of usually grasses. So they have asked the wrong statement. They are minute specialized subepidermal cell that occur on the not abaxial side, which is adaxial side of the leaves in Monocotyledonous plants, that is those belonging to Poaceae, that is grass family, not in dicotyledonous plants. So that is the correct answer. Then which of the following statement is incorrect? Again, incorrect statement. In meiosis, one cycle of DNA replication is followed by 2 rounds of chromosome segregation. That is correct. And it is known as the meiosis 1 and the meiosis 2. So, one cycle of DNA replication, 2 rounds of chromosome segregation, meiosis 1, meiosis 2. The natural type of mitosis is present in majority of animal cells. Yes. 2 homologous chromosomes in meiotic prophase 1 are held together by synaptonemal complex. That is also correct. Pluripotent cells have the ability to regenerate an entire organism in vitro. No. Pluripotent cells, they have the ability to differentiate into many different cells, not the ability to regenerate in an entire organism in vitro. That is the characteristic feature, that is regenerate an entire organism in vitro is a characteristic feature of the totipotent cells, not pluripotent cells. So answer here is option D. Then next one, which of the following compounds specifically cleaves peptide bond after methionine residue? So it is your cyanogen bromide, and you can see that in option C. Then FDNB, that is 1-fluoro-2,4-dinitrobenzene, it is used to modify amino groups. Trypsin, it cleaves the peptide bond at the of the basic amino acids, that is lysine and arginine. Then pepsine, it cleaves in a non-specific manner. So answer here is option C, that is cyanogen bromide. Then which of the following statement is not correct for chrysolaminarin? And chrysolaminarin, it is a polysaccharide, not a lipid, and it is the reserve food of Xanthophyceae. So chrysolaminarin is the reserve carbohydrate of Xanthophycophyta. That's correct. It is a neutral lipid triglycerol. That is not correct. It is a carbohydrate. So option B is the answer. It is a soluble polymer consisting of glucose monomers linked by beta-1,3 bond and limited beta-1,6 branching. That is correct. It is stored inside the cells, being encapsulated in vacuoles. So that's also correct. Then next one, the chemical nature of reserpine obtained from the plant Rauwolfia serpentina. It is an indole alkaloid. Answer is option B. Then next one, a molecular probe used in nucleic acid hybridization process is generally. So probe, it is an oligonucleotide sequence, and it is complementary to the single-stranded DNA which you are interested in. So it is a fragment. It is an oligonucleotide, means it can be a fragment of DNA or an RNA, and it is used to detect the presence of a specific nucleotide sequence in your DNA, and it is complementary to the target DNA sequence. Okay. So options, an oligonucleotide double-stranded DNA segment. No, it is not a double-stranded segment. It has to hybridize to your target sequence. So naturally it has to be single-stranded. So it cannot be double-stranded. So that is not correct. A large single-stranded polynucleotide fragment having random sequence of nitrogen base. It cannot be random sequence. It has to be the specific sequence. That is, it has to be complementary to your target sequence. So it should be specific sequence of nucleotides. A large double-stranded DNA fragment with random sequence of nitrogen base having unlabeled, again a random sequence, and unlabeling means naturally you will not be able to detect it. So that's also not correct. An oligonucleotide with the complementary, an oligonucleotide complementary single stranded DNA having labeled atoms. That is more correct and more accurate also. So that is the answer. Then which of the following statement is not correct for heterocyst? Okay, heterocyst, you all know it is involved in nitrogen fixation and is found in some cyanobacteria, and they lack, these heterocysts, they lack photosystem 2, that is they do not produce oxygen during photosynthesis, and it is a mechanism by the cyanobacteria, because if oxygen is produced, then the enzyme that is responsible for nitrogen fixation, that is your nitrogenase, would get inactivated by the presence of this oxygen. So they don't have pigment system 2 which produces oxygen during photosynthesis. So now read the statement. Heterocysts are specialized nitrogen fixing cells produced by some filamentous bacteria. That is correct. They have asked for the not correct statement. The presence of photosystem 1 ensures that sufficient ATP can be generated in heterocyst. That is also correct. The presence of photosystem 2. Photosystem 2, I told you, it is absent in heterocyst. Also ensures that sufficient oxygen is liberated in the heterocyst. That is wrong. I told you if oxygen is liberated, then the enzyme which is involved in nitrogen fixation will get inactivated in the presence of oxygen. So C is the answer. They have asked for the not correct statement. Heterocyst fix nitrogen from dinitrogen in the air using enzyme nitrogenase. That is also correct. Then next one. Gel filtration chromatography is a versatile method for efficient separation of biological molecules. Which of the following statement is not true for this chromatography method? So gel filtration chromatography, you all know it is also called as size exclusion chromatography, and it separates molecule based on their size. So the molecules are passed through a column filled with porous gel matrix, and the larger molecules, they do not enter the pores of the matrix and they will pass through the column quickly, whereas the smaller molecules, they will enter the pores and they will come only later. So it is a form of partition chromatography used to separate molecules of different molecular size. That is correct. This technique has also been referred to as gel permeation chromatography or molecular sieve chromatography. That's also correct. In this method, molecules are separated based on their affinity to the ion exchanger. No, that is based separate molecule based on their size, not on affinity. Affinity to the ion exchanger is for your affinity chromatography or ion exchange. Yeah, it is ion exchange chromatography. Then gel filtration matrices can be made from dextrans or agarose. That is also correct. Then next one. Wastewater treatment is a multi-step operation employing a number of independent processes. Which of the following stages of wastewater treatment is carried out by aerobic microorganism? So it is the activated sludge system. They use aerobic microorganism to break down the organic matter in the waste water. So primary treatment, it involves screening and sedimentation to remove large objects, suspended solids from the wastewater, and they do not use any aerobic microorganism. Secondary anaerobic wastewater treatment, they use anaerobic microorganism, not aerobic microorganism. Then tertiary treatment, to remove contaminants from waste water, and includes the use of aerobic microorganisms. That is, only some tertiary treatment processes involve the use of aerobic microorganism, but aerobics activated sludge system is the more correct answer. Then which of the following methods is used for protein detection using antibody based probes to obtain specific information about target protein from complex samples? That is your Western blotting. Southern blotting, it is for your DNA detection systems. Northern is for RNA. There is no Eastern blotting as far as I know. Then which of the following neurons in CNS have GABA receptor containing the alpha 6 subunit? So it is your granule cell that is having the GABA receptor containing alpha 6 subunits. Purkinje cells have GABA receptors, but they do not contain alpha 6 subunits. Then mossy fiber efferent, they do not have GABA receptors. Red nuclear efferent are also are not having GABA receptor containing alpha 6 subunit. Then next one. In signal transduction, the rhizobium establishes its contact with the leguminous plant through. So for nitrogen fixation, rhizobium is going to establish contact with the leguminous plant, and it is through surface polysaccharides of bacterial cell wall. Okay. Then the first transgenic cow developed in 1997 was Rosie. Which of the following genes was introduced in Rosie? It is human alpha lactalbumin. Then next one. Histone acetylation increases the transcription of gene because. So histone acetylation, it is a post-translational modification, and it involves the addition of acetyl group to the lysine of histone proteins. So this upon addition of this lysine residue to histone protein, it reduces the positive charge on the histone protein, and it will loosen the interaction of histone proteins with DNA. So as a result, what happens is the chromatin structure will become more accessible to transcription. So it will allow the binding of RNA polymerase. So that's what is happening upon histone acetylation, and thereby increasing transcription of gene expression. It increases the DNA histone interaction. No, it decreases it. The acetyl group of histones are recognized by RNA polymerase. No. Histone acetylation loosens the DNA histone complex, making it more accessible to RNA polymerase. So answer is option C. Histone acetylation increases the DNA bending, which is recognized by RNA polymerase. So that is also not correct. Then the Mycoplasma species are characterized by all of the following general features except. So Mycoplasma, they do not have a cell wall. So they are resistant to the beta-lactam antibiotics such as penicillin, because penicillin, they work by inhibiting the cell wall synthesis. So Mycoplasma, no cell wall, no action with beta-lactam antibiotics like penicillin. So species are characterized by all of the following features except. They form fried egg colonies on solid media. Yes, that's correct. They are wall-less microorganisms and are pleomorphic in shape. Yes. Penicillin is the drug of choice to control mycoplasma infection. No, that is the correct answer. They are osmotically fragile and related to bacterial L forms. Yes. Then which of the following enzyme is not found in lysosomes? So lysosomes, they are suicidal bags. So naturally they will have phosphatase, lipase, protease. But resolvase will not be there because resolvase is an enzyme involved in DNA recombination and repair. So it is not involved in the breakdown of cellular waste, that is resolvase. So answer is resolvase. The synthesis of mono unsaturated fatty acid palmitoleiate 16,1 delta 9 from palmitic acid is catalyzed by fatty acyl CoA desaturase in an oxidative reaction. The enzyme is, it is a mixed function oxidase. Then two extracellular domains present in each chain of T cell receptor heterodimer belonging to immunoglobulin superfamily domain are constituted of. So it is antiparallel beta sheet that are found in the two extracellular domains of the T cell receptor. Then which of the following organic groups have least probability of hydrogen bond formation with water? Now hydrogen bonding, it is formed between hydrogen atom that is bonded to the highly electronegative atoms such as oxygen or nitrogen or fluorine. Okay. So methyl group out of these, that is your answer, because that is having the least probability of hydrogen bond formation with water. It is hydrophobic and it does not have the ability to form hydrogen bonds with water. Okay. Because methyl group, it does not have highly electronegative atoms such as oxygen, nitrogen, fluorine that can participate in hydrogen bond formation. Whereas amino group can form hydrogen bonds with water through lone pair of electrons on the nitrogen atom. Hydroxyl groups can form hydrogen through lone pair of electrons on the oxygen atom, and carbonyl groups can form hydrogen bond through lone pair of electrons on the oxygen atom. Then next one. In symbiotic relationship which plants, which of the following is formed by ectomycorrhizal fungi? That is Hartig net. So it is a network of hyphae that surround the root cell, and it plays a key role in the exchange of nutrients between the fungus and the plant. So the fungus, it provide nutrients such as nitrogen and phosphorus to the plant, and the plant provides carbohydrates which are produced during the photosynthesis to the fungus. Then nodules, they are formed by rhizobia in relationship with leguminous plants, and they are site of nitrogen fixation. Then hostoria are formed by parasitic fungi, so they are also not correct. So arbuscules are formed by arbuscular mycorrhizal fungi and not ectomycorrhizal fungi. So Hartignut is the correct answer. In a stirred tank bioreactor, the primary function of impeller is. Okay, they have asked the primary function of impeller. Of course, it can be used for continual mixing of microbial cells in the liquid culture medium, but the primary function is to supply adequate oxygen to the growing culture. Answer is option D. Then measuring dissolved oxygen in the fermentation medium, it is done by the probe or the sensor. Then prevent the formation of foam in the vessel, impellers can break the foam also. But the primary function is supply adequate oxygen to the growing culture. Okay. Next question. In April 2016, the National Green Tribunal suspended the environmental clearance given to the proposed hydropower project in Arunachal Pradesh to save which of the following animal species? It is black-necked cranes. Then bacterial ribosomes are composed of two asymmetric subunits. The large 50th subunit of Escherichia coli ribosome consists of. So the 50th subunit of the bacterial ribosome, it is composed of 16S rRNA and 34 proteins. Answer is option C. Then Loktak is one of the 37 Ramsar sites in India. It is home for bro-antled deer. Then which of the following statement is most appropriate about Y chromosome in mammals? Essential for the development of embryo. It is Y chromosome, it is important for male development but not much involved in embryo development. Non-essential for the development of embryo but important for male sex determination. That is also not correct. Non-essential for sex determination but important for male sterility. That is also not correct because Y chromosome is important for the sex determination. So they have mentioned as non-essential for sex determination, which is not correct. Then essential for male sex determination and male fertility, that is correct. So option D is more correct than non-essential for development of embryo but important for male sex determination. In B, they have mentioned only about male sex determination, but it is also involved in male fertility. So D is more correct than B. Then which of the following incidents is not associated with Lyon hypothesis? So Lyon hypothesis, it is X chromosome inactivation, and in females one of the two X chromosome is inactivated, and this is to avoid a doubling of gene expression. So the inactivation of one X chromosome, it ensures that genes on the X chromosome are not overexpressed in females, because females have two X chromosomes whereas males have only one X chromosome. So that is the purpose of Lyon hypothesis. Then the inactivated X chromosome forms what is called as the bar body in females. Okay, now see the options. Consanguinity, that is marrying or members within the same family, that is marrying your own cousins. So it can lead to increased risk of genetic disorders, and it is not associated with Lyon hypothesis. So they have asked not associated. So answer is option A. Dosage compensation. Yes, I told you. Formation of tortoise shell pattern in fur coat of female cats. Yes. Because inactivation of one X chromosome in each cell lead to formation of different colors or patches on the fur of female cats. Because the genes which control the coat color in cats are located on the X chromosome. Presence of bar body in female cats, that is also correct. Then next one. What would be the average packing ratio of DNA in the solenoid structure of chromatin, that is 6. So in a solenoid structure of chromatin, the DNA double helix, it is wrapped around a core of histone proteins. So that forms what is called as the nucleosome, and this multiple nucleosomes, they come together and they coil together and they form what is called as the solenoid structure. So it is the average packing ratio of DNA in solenoid structure of chromatin is 6. And 146 number, if you remember, it is the number of base pair of DNA that is wrapped around the histone in the nucleosome. Okay, then the interaction in which individual gives up or sacrifice some of its own reproductive potential to benefit another individual, it is called as altruism. Okay, so it can include actions like sharing food, protection from enemies, helping in taking care of the young ones. All these are altruistic behaviors. Then the Hilsa shad Tenulosa ilisha is a migratory fish. It comes under the category of. So it is amphihaline potamotocos fish. Now what is all these words? So amphihaline means it can live both in freshwater and salt water. And potamotocos means the fish that migrates from the ocean to fresh water to spawn. That is potamotocos fish. So what is oceanodromous fish? That is a fish that is found in, it will migrate in the ocean only, it will not enter fresh water. Then potamodromous fish, it is a fish that migrate within freshwater environment and it will not enter the ocean. Then amphihaline thalassotocos fish, it is migrate from freshwater to ocean to spawn. So that's it with Hilsa shad. Then next one. The excretory products of earthworm are. So ammonia, urea and creatinine. Answer is option D. Then next one. A researcher was observing tail regeneration of planaria injecting with the RNAi for beta-catenin mRNA into the blastema. So RNAi is it will disrupt the normal functioning of the beta-catenin mRNA. Okay, what would be the expected result of his experiment? Planaria with an extended tail. No, planaria with heads at both the ends. Now beta-catenin, it is involved in WNT signaling pathway and it is required for the specification of head-tail axis during planarian regeneration. Okay. So when beta-catenin is disrupted, the normal head-tail polarity is disrupted and it lead to the formation of head at both the ends. So in why there is not two tails, why it is head at both the ends? Because in planar ends the WNT beta catenin pathway is normally active at the posterior end, that is it promotes tail formation. When beta catenin is disrupted, the pathway becomes active at both the ends, leading to the formation of head at both the ends. Okay, so a researcher was observing tail regeneration of planaria injecting RNAi for beta catenin into the blastema. So what would be the expected? It will get heads at both the ends because beta catenin is normally active at the tail end. So if you supply or inject RNAi for beta catenin mRNA, it will disrupt the beta catenin function at the tail end. And it will lead to the formation of heads at both the ends. So next one. During development in placental mammals, the fertilized egg undergoes division to form a blastocyst. The outermost cell of blastocyst differentiate to form a thin layer of cell which then invade the uterus and begin to form the placenta. This thin layer of cell is known as the trophoblast. Answer is option D. Then excretory organs of herdmania is solenocytes. Answer is option C. So coming to the next question. Which of the following environmental pollutants cause methemoglobinemia in humans? It is nitrate. Then according to the pressure flow model, mass flow of solute from source to sink occurs due to. Now the source is your leaves that produce sugars and other solutes during the process of photosynthesis. Then these solutes, they are loaded into the phloem tissue, thereby increasing the turgor pressure within the phloem tissue. Now this increased turgor pressure, it creates a turgor pressure gradient and this will drive the solutes from the phloem tissue towards the sink tissue. So according to pressure flow model, mass flow of solute from source to sink occurs due to turgor pressure gradient. Answer is option B. Congo jute, an important superior jute substitute, is produced from Unina lobata. Answer is option B. The chromosome number of antipodal cells of a cereal plant is 10. What would be the chromosome number in the cells of alluron layer of the grain of this plant? So the antipodal cells, you all know, they are haploid. So n is equal to 10. So alluron layer, it is the outermost layer of the endosperm and endosperm is triploid. So 3 into 10, that is 30. So your answer is option C. So coming to the next question. Which of the following pump triggers pollen tube growth and discharge in the event of double fertilization? It is your calcium pump. Answer is option C. Then next one. The spirochetes form one of the major phyla of the eubacteria and are characterized by the following features except. Morphologically, most spirochetes have a long helical coiled or screw shaped cells. So that is correct. Like gram-negative bacteria, spirochetes are diderms or double membrane bacteria. Yes. Spirochete flagella are referred to as endoflagella or axial filament. Yes. So that's correct. They have asked are characterized by following features except. All the spirochetes are free living and can be cultivated from virtually any moist, nutrient rich environment. All the spirochetes are free living. So that's not correct. All spirochetes are not free living. There are parasitic and symbiotic ones also. So option D is not the right answer. Then next one. Primary production in an aquatic ecosystem is usually measured using light and dark bottle technique. In this method, the dissolved oxygen concentration of the pond water enclosed in BOD bottle before incubation and after a fixed duration of incubation in a dark bottle and light bottle were measured. Then the gross and net primary productivity of this system may be estimated as. So BOD is biological oxygen demand, the BOD bottle is filled with pond water and it is incubated in both light and dark conditions. Then the dissolved oxygen concentration is measured before incubation and after incubation in both the light and the dark bottles. Okay, now you have to find out what is the gross and the net primary productivity. Now gross primary productivity is the total amount of organic matter that is produced by photosynthesis. So it is estimated based on the difference in the dissolved oxygen concentration between the light and the dark bottles. So it is capital L minus capital D. That is gross primary productivity. Now what is this net primary productivity? And it is the amount of organic matter produced by photosynthesis minus the amount lost due to respiration. That is net primary productivity. So it is estimated based on the difference in the dissolved oxygen concentration between the light bottle and before incubation, that is I. So L minus I. So the answer here is option A, that is L minus D and L minus I. So that's it. Thank you for watching my channel. Please do like, share and subscribe if you are new to my channel. If you want still previous year question papers of West Bengal set been explained, you can go to my channel Omni Bio Entrance. In the channel playlist, you can go to WB set and there you will find. Thank you.